Swayam Prabha. Digital India. Educated India.
Good day everyone, welcome to the course on next generation sequencing technology, data analysis and applications. In the last class we have started looking at the SAM file format and we have described the header section, right. So, we have seen what kind of information you have in the header section. So, in this class we will be now going deeper and we will be looking at the alignment section where actually we have the most interesting results, right. So, where the mapping results are reported. So, that is something which we will cover in this class SAM file format. So, these are the keywords that will come across something called mandatory fields, flag and SIGA. Okay, so, just to summarize what we have seen is that we have we know the full form, right. The SAM stands for sequence alignment map, and BAM is the binary version of that file. This is the machine readable file, and there are two sections, header section and alignment section. Now, in case of BOTI2, so by default the BOTI2 will print the SAM header with @HD, @SQ and @PG. Okay, so, this information will be there in the header section. Now, sometimes what you will see, right, if you have a lot of information in the header section, it might be difficult to actually look at the alignment section. Okay, so, what we can do is that we can remove this header section when we are running BOTI2. Okay, so, BOTI2 gives us that option as well, and you can do this by this running these options minus no head, minus no SQ when you are running BOTI2. Okay, and we can do that right before we actually start going into the alignment section, because the header section could be a bit distracting. So, let's do that. Let's run this and then we can see right how this will change the output file. Okay. So, we are going back to the original command, right. We have this we are supplying this iteration 1 trim data, the trim read 1 trimmed and read 2 trimmed data, right, and then we will give this option. We can have this minus local option, and then we can give these options minus no head and minus no SQ. Okay. So, we do not want this header or SQ lines. So, we can simply use these commands to remove those. Okay. Okay, so, it will run and we will see the output file, and you will see there is no header line, and it looks clean, right. So, in some cases this might be useful for you. Okay, because if you are doing the alignment yourself, right, you know what this reference genome is, right, what is the length of this reference sequence and which program you are using, etc. These are known to you. So, you are not getting any new information, right. So, maybe having this header line does not make sense in your case. Okay. So, let us look at this output file now once we have removed this header, and you can see we come in the alignment section straight away. Okay. We do not have any header, right. So, you can go to the first line of this file using this :1 command in VI, and you can see there is no header. Okay. We are looking at the alignments. Okay. And we will now try to interpret this data. Okay. You see a lot of information in this file, and we will kind of interpret what this information means. Okay. And this is very important if you want to write the codes yourself to process these files. Okay. So, we will go back to the presentation and I will introduce these terms, and then we can come back to this file and have a look. Okay. What kind of information we have in the actual results, and how do you interpret that result, right. Okay. So, the alignment section, right. So, in alignment section you have these multiple lines that you have just seen, okay, and each line describes an alignment for a read, or it will tell whether there is a failure. Okay. So, if a read did not align, it will also say that in that alignment file, alignment section. Okay. And one thing we should remember is that alignment of all map reads are represented on the forward genomic strand, right. So, this is by convention, and if you have reads that map to reverse strand, so in this alignment section they will be provided as reverse complemented. Okay. So, when you are interpreting the data, you should keep that in mind. Okay. So, as I said, each line contains alignment of one read, and it is a collection of 11 or more fields. Okay. And these are all separated by tabs. Okay. So, this information is important, because if you are trying to process these 5 yourself, you will use this tab separation, right, to get those fields. Okay. Now, why did I say that we have 11 or more, right? Why not a fixed number? Because in SAM format you need to have mandatory fields, right. So, these will be there always, and these are the first 11 fields. Okay. So, you will have at least 11 fields, these mandatory fields will always be there, and on top of that you have something called optional fields, right. So, these may or may not be there in the file, and you in some cases you will probably see them. Okay. So, in our data you will see these optional fields, these are there because they are also giving us some important information about the mapping. Okay. So, what are these 11 fields? So, I will go one by one, and we will also interpret what actually these fields convey. Okay. So, some of them are really straightforward, so you probably understand very easily, some of them are not, right. So, that I will elaborate those fields a bit more, and you will probably understand what actually those mean. Okay. So, let us move into the mandatory fields first, right. So, and then we can discuss the optional fields later. Okay. So, the field 1 in the compulsory or the mandatory fields is something called Q name, right. So, this is how we denote this by capital Q name, right. So, all in capital, right, and this actually mentions the name of the aligned read, right. So, this name actually comes from the FASTQ file, right. So, you have seen that we have some header and we have name of the read, okay, and that is copied in here. Okay. So, we can actually now tally, right, between the FASTQ file and see which read actually mapped where. Okay. So, this information is there, and this information is also important, right, if you are dealing with paired end data. Okay. So, reads with identical Q name, they are assumed to come from the same template or fragment, right. So, for paired end data they are coming from the same fragment, right, we are sequencing from both direction. So, the Q name would be identical, and we have seen, right, if when you are dealing with this read 1 read 2 files, right, there are two different files, read 1.FASTQ and read 2.FASTQ. If you look at the header names, the names are identical except a second field, right, where the read number is given, right, 1 or 2, right. So, that is not part of Q name. Okay. So, that second part of the header in FASTQ file is not part of Q name. Okay. As you will see in a moment, right, the Q name consists of the first part of the header in FASTQ file. Okay. So, it simply tells us about the name of the aligned read. Then you have the second field, which is called flag. Okay. So, this is a combination of flags, which is like a sum. We usually take the sum of the flags that are present. Now, you might ask, what are these flags? Okay. So, they are actually describing certain characteristics of the read that we are dealing with. Okay. So, it is not just the characteristics of the read, but in addition it also describes characteristics of the mate in case of we are dealing with paired end data. Okay. So, let us look at these numbers, right. So, they are denoted by usually by numbers, and we take sum, the final flag that we report in the SAM file or the program reports is a sum of these numbers. Okay. So, what we will simply do is we will look at these numbers, and you can identify certain cases where these number would be applicable for your read, and you simply take those numbers and sum them up. We will take some examples also for a better understanding. Okay. So, the first flag, right, which is denoted by this number 1, this is the read is one of a pair. Okay. So, we are kind of trying to interpret this in the context of Illumina data. Of course, this is there is slightly different interpretation or slightly general interpretation of these flags. Okay. So, for example, you will see this one does not always mean paired end data, it means you have templates matching to multiple regions of the reference. Okay. So, we will kind of interpret this in terms of Illumina data, because we are kind of going through examples of Illumina data. Okay. So, flag 1 will be applied when a read is one of a pair, right. So, now you can imagine if you are dealing with single end sequencing data, you will not apply this flag on your read. Okay. Then you have flag value 2, which is the alignment is one end of a proper paired end alignment, again applying to paired end data, right. So, this will not be applied in case you are dealing with single end data. Then you have flag 4, not 3, okay, flag 4, and it is applied when the read has no reported alignments. Okay. This is something that will apply to single end as well as paired end data. Then you have flag 8, right. So, not 5, 6, 7 in between, those numbers are not there. You have flag 8 and the read is one of a pair and it has no reported alignments. Okay. So, flag 4 and flag 8 they are slightly different, right. As you can see, flag 4 will apply to both single end and paired end, whereas flag 8 will apply only to paired end data, right. Now, flag 16 is the alignment is to the reverse of the reference sequence, right. So, reverse of the reference strand. Okay. And then you have 32, which actually is describing now the alignment of the mate. Okay. So, 16 is the alignment of the read that you are working with, right, or the read for which you have the Q name. This value of the flag is for the mate. Okay. So, the mate or the pair is in the paired end alignment is aligned to the reference strand. Okay. So, it kind of gives you the orientation, right, which one is occurring upstream of the other one. Okay. So, this information kind of is given here. Then we have flag 64, which means the read is mate 1 in a pair. So, it is giving us information about these mates, right. So, mate 1, mate 2. If we have flag 128, this means the read is mate 2 in a pair, right. So, we are dealing with paired end data, we will get these flags. Then we have 256. So, it will tell us whether this is a primary alignment or secondary alignment. So, if you see this flag, this means that this is a secondary alignment and there must be some primary alignment that has been described. Then you have 512, there is some sort of quality filtering or this read is not passing this quality control check. Then you have 1024, which means this is a duplicate PCR or optical duplicate. We have described what is PCR duplicate or what is optical duplicate. Then you have 2048, it means there is a supplementary alignment or chimeric alignment. Now, these chimeric alignments happen primarily due to structural variation or gene fusions. So, if you can imagine, right, let us say 2 chromosomes are fused, right. So, one part of the read will map to let us say chromosome 1, the other part of the read will map to chromosome 10, right. So, if those kind of situation happen, then you will get this flag 2048. Now, one thing you would notice that these flags, right, one in the earlier slide as well as here. So, they are actually kind of separated, right. So, you have these 2 to the power 0, 2 to the power 1, 2 to the power 2, so on, right. This is done to actually generate unique sums, right. So, as I have mentioned, this flag that we get in field 2, this is actually sum of applicable flags. So, the sum should be unique, so that you can figure out which flags were applied on a read. Have these numbers, right, and they are not in consecution. So, why is that, right? Because when you take sum for each of these unique combinations, you would get a unique sum value. Okay. So, that is why these are followed in this way. Okay. And let us take some examples, so that we understand how we calculate these flag values. Okay. So, the first one is, let us say I give you a scenario and then you have to identify what is the flag value. So, the example 1 is an unpaired read that aligns to the reverse reference strand. Okay. So, I have mentioned this unpaired read and it aligns to the reverse reference strand. Okay. So, you do not have to of course remember the flag values, just looking at the table you have to kind of now deduce which kind of flag you would apply on this read. Okay. So, this is an unpaired, right, data. So, you will not apply any of these paired end data flags, right. So, only thing I can see, right, that is right. So, it aligns to the reverse reference strand, the only thing you can see is 16. Okay. So, that is actually would apply, this will apply to single end data as well, and the flag is 16. Okay. So, you can try this yourself, you can go back and see the table, right. I am not seeing this in front of me. So, slightly difficult to go back and forth, but you will see once you look at the table you can see which kind of, if you have this tag applicable for your data, then you can apply that and just take sums of all the flags that are applicable. Okay. So, let us take a second example where you can apply multiple flags and that then it will be clear, right, what is the final flag. Okay. So, this is a more interesting situation, right. We have a paired end read that aligns to the reverse reference strand and is the first mate in the pair. Okay. So, there is a lot of information, it is a paired end data, means there will be a lot of flags that could be applicable, right. It also it gives that information that this read aligns to the reverse reference strand. Okay. So, if you go back to the table, I am not going to do that, but you can check the table and you can apply the appropriate flags and you will see the flag will be 83, which will be 64 plus 16 plus 2 plus 1. Okay. So, all these flags, this 64, 16, 2, 1, these flags are applicable in this situation for this read, and you just take sum of these and you get this flag value of 83. Okay. So, you can now actually do that yourself, right, and you can interpret the data, and the sum will be unique depending on the combinations that you use. Okay. So, what I will do, we will now look at the SAM file and we will look at these first few fields that we have just described, okay, and especially the flag field, we will see what kind of value we get in the actual data. Okay. So, let us go to the terminal, right, and let us look at the output file. Okay. We open that output file using this VI, right, and we use colon sc no wrap, right. We remove the wrapping because we can kind of see this very nicely, the data organized very nicely. On the first column you have the queue name, right, this is the read name, right. And another thing you probably would notice here, these first two, right, these they have same queue name, that is because they are coming from the paired end data, right. So, they are read 1 and read 2 of the same fragment, and so you can see this data is organized in pairs, right. So, you have these first two, then the next two, and so on, okay, because we are dealing with paired end data, this is what is expected. The second field, as I have mentioned, this is the flag, right. Here you have this 99, 147 and you see this 83, right. We just described how you get this flag 83. So, you see this flag 83, then you have 163, etc. So, again depending on the situation, right, you can calculate this flag value, which will be sum of all the applicable flags for the read that you are working with. Okay. So, this kind of gives you the gives you a feel for the real flag values that you have. Okay. We can go down and you will see you have only few combinations around, right, and that is generated by the method that we just described. Okay. So, we can now go back, we will keep this open, but we can go back now to the presentation and look at the other fields after this flag. Okay. So, we have described the field 1 and 2. Now, we can go back to field 3, right. So, field 3 actually describes now the something called R name. Okay. So, this is the name of the reference sequence where alignment occurs. Okay. So, reference sequence means if you, depending on the data whether actually this is stored as chromosomal data, right. So, it will give you the chromosome name, right. So, in the FASTA format file that we used in the mapping process, we have the chromosome name or chromosome number or chromosome ID, some sort of ID. So, here in R name it will just give that chromosome ID, and then you will also have a field that will give you the position, right. So, what you will see is that if you have this @SQ header line. So, we have removed that now, but you have seen this R name will be one of the values. Okay. It is there. So, this is the sequence name. So, as @SQSN it will be mentioned, this R name value will be mentioned. So, if it is let us say chromosome 1, you will have a header line saying @SQSN chromosome 1, right. You have seen this example that we have just that I just showed you, right, with the header. Okay. So, in addition you also will have this @SQLN, right. It was there and that was giving the chromosome size, right. So, that will give you the chromosome size, length of the chromosomes, etc. Okay. So, for unmapped reads you will not see an R name, right. So, because there is no mapping, so the R name will have a value of star. Okay. So, if you see the star means this read did not align against the reference sequence. Okay. So, after this reference name what is expected is the position, right. So, you want to know the position in the reference sequence, and that is what field 4 gives you. Okay. So, this is the something called POS or the position, and this is a one based positioning system in the forward reference strand. So, as I mentioned when I started with the alignment section, I just mentioned all alignments are given with respect to the forward reference strand, right, and this is a one based positioning system. So, I will explain in the next slide what is this one based position system. Okay. and it gives you the location where the leftmost character of the alignment occurs. Okay. So, in the reference sequence where the leftmost character of the read occurs in the reference sequence, that is the position that we get in this POS. Okay. And this is set to 0 if you have unmapped reads. Okay. So, if your read did not align, you will get this to 0. Okay. So, just to clarify, right, this is what you will get, this is the position. So, if you have this read 1, read 2, if you are describing the results for read 1, it will give you this position, right. So, the leftmost position here. Okay. All right. So, here is the position, right, this position will be given in the POS field. Okay. So, this is the fourth field. Now, you might ask, like, what is this one base positioning system or one base coordinate system? Okay. So, this is a coordinate system where the first base of a sequence is numbered 1, right. So, you might be kind of wondering why we have to explain this, why 1 based and so on, because there is something called a 0 based coordinate system that we use in many cases. Okay. So, this is something that we have to explicitly clarify, right, because otherwise you might have problem with interpreting the exact location of this read, right. So, you might actually make some mistakes there. So, this is a coordinate system where the first base will be numbered 1, okay, and this is denoted in, if you are working with this kind of one base system, it will be denoted by these closed brackets. Okay. So, for example, if you have a region between 5 to 9 base inclusive, this will be denoted by this 5 9 closed bracket. So, notice this one here, because you will see in case of 0 coordinate system, this will not be closed, this will be a different sign. Okay. And this SAM file, VCF formats and GFF formats. So, these are VCF and GFF, these are other formats. So, for example, we will come to VCF format later on and GFF format also when you talk about gene ontology. So, they use this one based coordinate system. Okay. So, it is good to know that these files use one based coordinate system, because if you when you are using these files, you need to keep that in mind. Okay. And as I mentioned, there is a 0 based coordinate system where the first base of a sequence is numbered 0. Okay. Instead of starting from 1, you start from 0, and if you are familiar with programming, we see, like, in some cases we actually start counting with 0, not with 1. Okay. So, this is where this system uses the same kind of idea, you have 0 based system. Okay. And if you have this, right, in your system, you have this between the same example where between 5th and 9th base inclusive, in 0 based system this will be denoted by 5 9 with this open bracket, okay, not the closed bracket or the third bracket. Okay. And then BAM and BED formats as well as there are other ones, right, they use this 0 based coordinate system, right. Again, this is important to know if you are working with these files and you are writing codes to extract data from this. Okay. So, the field 5, right. So, we have now got this query name, we have got the flag, right, which kind of describes some of the properties of the alignment, right. Then we have got the reference sequence name, we have got the position information. Then the field 5, it gives you something called map Q or mapping quality. Okay. So, it is actually comparable to what we have described before for base quality, right. So, very comparable idea. So, you get a number in mapping quality value, and this is equal to minus 10 log 10 E. Okay. So, it is kind of very similar idea, like we calculated probability of error for a base, right, given the quality score, it was 10 to the minus q by 10. It is a very similar idea here. So, here E denotes the probability of error in the mapping process, right, instead of base call, this is a probability of error in the mapping process. So, this denotes the probability that the mapping position is wrong. Okay. So, this quality score, given the quality score, right, we can calculate what would be the probability of error, right. So, higher quality would denote, right, okay, we can be very confident about the alignment and it is probably not a wrong mapping. The value is set to 255 if the mapping quality score is not available, right, the program does not calculate this mapping quality score. Okay. So, Botai2 actually does calculate this mapping quality score map Q value, and we will see that. Okay. So, before we go into field 6, what I wanted to show you is the data, right, for actual data how for these fields that we have just described. Okay. And then we can come back to the field 6, which actually stands for SIGA. So, this is a SIGA string representation, and again it is quite complex information, and I will describe what this actually mean. Okay. So, let us go back to the terminal and see these fields that we have just described. So, first two fields we have, I have mentioned already, you have query name, and then the flag. The third one is the RNAME field, right. So, here is this reference NC some number, this is actually pointing to a chromosome ID. Okay. So, this is the ID that is given in the reference sequence, and that chromosome ID is given here. Okay. So, we can actually interpret, right, we can actually map it back to the chromosome, whether it is chromosome 1 or chromosome 6 in that organism, we can find that out using some mapping. Okay. So, this is RNAME, and then you have the fourth field. Okay. So, the fourth field gives you the position in that chromosome, all right. So, here you see, right, the fourth field. So, again for pairs you can find these locations. So, here is this 831-808. So, in this chromosome, in this position, the leftmost character aligns for this read. Okay. And you can see these numbers vary, right, for reads, and they are mapping to different chromosomes, but the pair usually would align to the same chromosome, right. This is kind of expected because most of these alignments will be concordant mapping. Okay. All right. So, these first 4 fields are done. The next one is the map Q. This is the mapping quality score, which is 44, right. So, this is quite reliable, right. So, as you have seen, right, we can calculate the probability of error in mapping by saying calculating this minus 10 log 10 E, right. So, if you can calculate you will see like higher value is better, right. So, here this is good, but for some alignment you see this value is 1, right. So, maybe these alignments are not that great, and perhaps we may have to quantify, or maybe we have to filter this out when we are processing these alignments. Okay. So, that is for later. If you are processing this file, you may have to be aware of this quality score. Okay. So, the next field I will just mention here, this is the Seeger string. Okay. We will now try to interpret what this actually mean. This is a combination of number and letters, and we will see how you actually interpret these results. Okay. So, here is the let us go back to the presentation and let us see what this Seeger string actually means. Okay. So, Seeger string consists of some numbers and some letters. So, in SAM file these will be denoted by letters. In BAM file these are represented in numbers. So, M stands for alignment match. Okay. So, do not confuse these with match, sequence match or mismatch. This means that nucleotide is present in the reference sequence. So, for a read you have a reference sequence base. Okay. So, this means there is a match or mismatch, and there is no indel. Okay. There is no insertion or deletion. Okay. So, here is the situation, right. You can see this will be described by match and mismatch. Okay. So, you do not have to you cannot interpret, right, whether there is a base mismatch or base match. What it simply tells, okay, that there is a base corresponding to the read in the reference sequence, and there is no insertion or deletion. Then you have the sign I in SAM format. This is 1 in BAM, and this is insertion, means the nucleotide is present in the read, but this is not present in the reference sequence, and this is the situation, right. So, this is an insertion in the read. Then you have D, which is the deletion. The nucleotide is present in the reference, but not in the read, right. So, this has been deleted in the read. So, that is why we call deletion, and this is an example, right. So, here you see this G present in the reference, but not present in the read. So, this would be a deletion. Then you have something called N, okay, which is the skip region from the reference. Okay. So, this is a region that is not present in the read. Okay. In case of RNA sequencing data, you will see this situation where we can interpret this N means there is an intron, right. So, you can have this kind of gapped alignment, right, or split alignment, where you have these two parts, right, two exons aligning to slightly different positions, right, slightly apart in the reference sequence, right, and you can have these N or the skip regions. Okay. So, in that case you will have these N values. Then you have this concept of soft clipping or S, right. So, what is this soft clipping? Okay. So, this will appear when you are doing local alignment, and these bases are not used for alignment. Okay. And this will appear mostly at the end of the read most of the time. Okay. So, this is present in the read sequence, but and also they are shown in the SAM file, right. They are part of the read sequence and also shown in the SAM file. Okay. Why do we worry about this? Because there is something called hard clipping, which is denoted by H. Okay. So, again this applies to local alignment, and these bases are not used for alignment, but then these sequences are also removed from the read sequence that is shown in the SAM file. Okay. There is a final concept which is called the padded alignment, right. So, this gives us an idea about how inserted sequence in the reads affect alignment against each other. Okay. So, this will take a bit of explanation, right. What is this padding, or what do I mean by padded alignment? So, padding gives us an idea about how inserted sequence in the reads affect alignment against each other. Okay. So, it is not comparing in the reference sequence, because in the reference sequence these bases are not present at all, right. So, let us take this example and then it would be clear. So, imagine this example where you have this reference sequence. So, star means gaps, right. So, there is no gap, there is no base in there. In the read 1 you have this insertion, right, G A, but in read 2 we see only one base A, we do not see G, right. So, we have this star, and in read 3 we do not see any of these inserted bases. So, we just see stars, right. So, if in SIGAR what we will show is that 8M, 2I, 9M, and so this means in the first read we have two insertions corresponding to the reference. In read 2, read 3, right, there is padding, right. So, the first base here in read 2, what you will see is that is not present, right. So, we can use something called 1P, this is a one base padding, then you have this one base insertion, right. In the read 3 example there is no insertion. So, this will be used as 2 base padding, right. So, 2P. Okay. So, we can actually kind of also look at this a bit carefully when you actually look at the data, right, how this actually is interpreted. Okay. So, we have again this equal sign, which means sequence match, and finally, we have X, which is called the sequence mismatch. Okay. So, well in the next class we will actually look at some of the actual SIGAR strings. So, I have just very briefly or very quickly showed you, okay, this is the column that shows the SIGAR string, but we will try to interpret in the real data in the next class. Okay. So, these are the reference that I used for this class, and then to summarize, we have now learned about the SAM file format. We have looked into the header section in the last class, and we have started looking at the alignment section. We have not completed because there are 4 more fields that are present, and we will describe those in the next class. And alignment section, as I mentioned, there are 11 mandatory fields that will always be present in a SAM file, and then you can also have some optional fields. Okay. And these mandatory fields, they describe mapping of a read and also about its mate in case of paired sequencing, and we will actually look at the second part in the next class. Thank you.